Yeah.
That's how this works. So, hello. Today we're going to focus on kind of the mechanisms of abnormal contractility and the role of the importance of uterine contractility in controlling bleeding after delivery. And then tomorrow we'll focus more on the actual treatment of uterine atony. So again, as Dr. Foley mentioned, all of this is kind of built, or this section is built around the concept of uterine atony. And uterine atony is, I'm going to propose one potential mechanism that seems to be associated with an increased risk of uterine atony, but clearly there's many, many, many more, and we can hope to discuss at the end of the talk some of those and where we can go from there. So we all know that there's a 50% increase in blood volume in pregnancy, and then that correlates with a corresponding 50% increase in cardiac output during pregnancy. The increases along gestation to a max in a normal weight woman of approximately 7.5 liters per minute. So here's the problem. Of that 7.5 liters per minute, approximately 20% of that goes to the uterus, and 80% of that to the placental bed. And when the placenta separates with a normal delivery, and we're not talking about a accreted or previous, but just a normal low-risk delivery that may be up at the birthing center up the street from your hospital, we have the potential of losing 1.2 liters per minute if all of the mechanisms aren't in line to control that bleeding. And so that's what we're going to talk about today. So we'll quickly just run through this. This is a very interesting article done by Dr. Smith. It was a review article in the New England Journal on parturition. And basically all I want to show is that there's this clear transition in the myocyte during pregnancy, where during pregnancy it's in this hyperpolarized state, and at the onset of labor it becomes highly depolarized, filled with intracellular calcium, and prostaglandin and the oxytocin receptors are highly important at then initiating this calcium within the higher levels of calcium within the cell, which then positively feed back on the SR and other ion channels. So you've got the cell full of calcium that's just ready to contract and allow the actin myosins to interact and lead to this contractile state. So uterine atony is a result of dysregulation of normal uterine contractions after delivery, and there's numerous risk factors. Here are three: prolonged labor, prolonged oxytocin augmentation during labor, high doses of oxytocin, anything that overdistends the uterus, and that may be a large baby, multiple gestations, so more than one baby in the uterus, polyhydramnios, and then we talked about, Dr. Foley had mentioned magnesium therapy, general anesthesia with fluorines, and all of these markedly increase your risk. A lot of this talk today, I'm going to focus on that these top three: prolonged labor, prolonged oxytocin augmentation, high-dose oxytocin during labor, have the potential to lead to desensitization of the oxytocin receptor. And once you have desensitization of that receptor, this is paradoxically then associated with decreases in uterine contractility. You think about oxytocin initiating this very, very strong contraction, either during labor or immediately postpartum. But in these settings, we have this issue where there's this paradoxical decrease. And this is the oxytocin receptor we'll talk about is a member of the G protein coupled receptor family. And as a family, they're very, very prone to desensitization, whether you're using opioids or whether you're using terbutaline for preterm labor. To kind of outline this, is a very interesting study done back in the early 90s. In this group, what they did is they put intrauterine pressure catheters in the uterus and measured the pressure generated on the y-axis. And in the second panel, they then started oxytocin, and we see an increase in the pressure generated. But when you kept the oxytocin infusion at that constant rate, after about an hour, we started seeing this slow decrease in the amount of force that the uterus is able to generate. They then doubled the rate to 2 milli-units per minute. We took about an hour because of the pharmacokinetics of oxytocin to get to a maximal contraction force that was generated. And guess what happened? A little bit later, we started seeing a fall in the amount of force that's generated when oxytocin is kept at that constant rate. And I propose that this is all secondary to desensitization, which we'll talk about. So we use some Duke data in this very, very small case control study to say, look, among women who are exposed to oxytocin, then we correlate the exposure to some adverse clinical outcome that's associated with abnormal uterine contractility. So we took women with uterine atony, women with postpartum hemorrhage secondary to uterine atony who received a blood transfusion and compared them to matched controls. And then what we did is measured the total amount of oxytocin that they received in labor, calculated that as this area under the curve, and not surprisingly, we see that those women with uterine atony, due to or postpartum hemorrhage due to uterine atony had markedly increased exposure to oxytocin. And when we went through the adjusted analysis and found that that association persisted, and the adjusted odds ratio was 1.58 in predicting uterine atony, and the exposure was a 5,000 milliunit increase in oxytocin, which is equivalent to being on oxytocin at 20 milliunits, which is a pretty classic dose for 4 hours. So for being on oxytocin at 20 for 4 hours, every 4 hours we had a 58% increase odds of uterine atony. We then went to the MFMU cesarean registry data, which we heard about earlier from Ware on the accreted data. The MFMU did not collect total dose of oxytocin, so they didn't have that area under the curve measurement. But we went through and identified risk factors, and we found that maximal infusion rates greater than 20 milliunits per minute during labor were an independent risk factor for atony in this data set. So in summary for this, prolonged or high-dose oxytocin exposure during labor has the potential is associated with decreases in uterine contractility in the postpartum state, which then increase the risk clearly for uterine atony. And I propose again that oxytocin receptor desensitization is a potential mechanism for all of this. So let's now talk about the uterine contractile phenotype. The uterus, as Evelyn had mentioned earlier, is an amazing organ. This organ that grows throughout pregnancy, it doesn't contract at all. The onset of labor, the phenotype is characterized by this phasic uterine contractile phenotype, and then guess what happens immediately at delivery? It immediately transitions to this tonic tetanic contraction to prevent postpartum hemorrhage. So during labor, uterine contractions are characterized by this phasic contractile pattern, and that's largely there to facilitate gas exchange to the baby. Oxygen coming into the placenta or CO2 leaving the placenta only occurs between contractions. In the postpartum state, the uterine contractile phenotype is characterized by this tonic tetanic contraction, which allows for these uterine placental pipes to be closed. And early in pregnancy, we have remodeling of the spiral arterioles that are responsive to vasoconstrictive agents, and they get remodeled and they lose that ability to be responsive to vasoconstrictive agents. So there's these huge pipes that are allowing blood flow to the fetus for 40 weeks, which is probably evolutionary mechanism, so that if you accidentally take your vasoconstrictive agent of choice, that you're not cutting off blood supply to the fetus. But the problem with that design is that in order to shut off that 1.2 liters per minute, you need adequate contractions around it to close it off. So the molecular mechanisms regulating this transition from that phasic phenotype to an immediate tonic tetanic contraction at delivery, we have no idea. And again, failure of that leads to uterine atony and postpartum hemorrhage. So here's just a classic phasic contractile phenotype. This is myometrium that's removed at the time of delivery and put in a tissue organ bath, and we can see that when you treat the strips with increasing doses of prostaglandin, which is a very potent uterotonic agent, we see increases in contraction frequency and amplitude. Now, I borrowed this figure from my good friend, Dr. Brancazio, who many of you probably know through this organization. But what this is that this is the wall of the uterus, the uteroplacental pipes bringing blood through the myometrium to the placenta. During a contraction, the intramyometrial pressure within the wall of the uterus is greater than the mean arterial pressure of those pipes. They shut down. So what that does is during every contraction in labor, there's no more oxygen getting into the placenta, no more CO2 getting out, and the fetus is just living off this reservoir of what's in there, and then when the contraction relaxes, you get it back out. So now here's a representative figure of that tonic contraction phenotype that we're ideally seeing after delivery. Now we go back to Leo's picture. The placenta is now off. We have these huge open pipes at 1.2 liters per minute, and ideally we get that tetanic uterine contractile phenotype that shuts down those vessels, stops that bleeding, and then allows all the hematologic stuff that you all are experts in to work at that site. But you first have to close the hole, and I can't remember who ever mentioned that in the last. So that allows the hole to be closed so that all the fancy proteins can do their job. So then the problem comes when some mechanism occurs where that contraction doesn't occur, and then we're again back to our 1.2 liters per minute. So how do we prevent this? So we know that there's clear risk factors for uterine atony. The problem is not all women with uterine atony have one of those identifiable risk factors. And we deliver women all the time at Duke and at Salt Lake and at Banner that have all of those risk factors and do completely fine. So what do? Well, oxytocin bolus has become the mainstay preventative therapy for all women who deliver in our hospitals. And we use this prophylactically, again, for all women. And what we do is that we give them a large bolus of oxytocin that's diluted in saline or LR. And this dose is probably 50 to 300-fold greater dose than is utilized in labor. And there's something about that high dose that allows this, that helps this transition from this phasic contractile phenotype to the tonic one. Clearly, women deliver fine without an oxytocin bolus, and they have that transition anyway, but the mechanisms that drive that we don't know, but boluses of oxytocin clearly help that transition, and we utilize it for all women. Okay, because of the importance clearly of oxytocin and ideally preventing uterine atony, and then the paradoxical link with prolonged exposure, we need to talk about oxytocin signaling. So again, the oxytocin receptor is a G protein coupled receptor. As a family, they're very prone to desensitization. When the receptor is activated, it couples to a G protein, and for this class of receptor, as with the prostaglandins, it couples to GQ, which leads to increases in intracellular calcium and all the magical contraction, and all the magic of actinomyosin in the cell. Simultaneously, as all that G protein signaling has occurred, the receptor is phosphorylated by this kinase called GRK6, which then allows recruitment of this protein called beta-arrestin, and beta-arrestin promotes desensitization of this entire class of G protein coupled receptors. And what it does is it immediately separates the receptor from its G protein, shuts off further downstream signaling, the receptor gets internalized into the cell, and then recycled back to the membrane where it's then available for the next contraction, and however long that recycling takes. So what we did to try to mimic oxytocin desensitization in the lab and the effects on contractility, we took myometrial strips from wild-type mice, hung them in a tissue organ bath, treated them with increasing doses of oxytocin, and measured the contractile response. And that's that black curve. So a nice increase in contractile strength. Then we took the strips and immediately retreated them with that same increasing dose of oxytocin. And what do we see? We saw the gray line, and the strips were desensitized. With that immediate retreatment of oxytocin, they generated less force than we saw with the initial exposure. Then we took mice that lacked key components of the desensitization pathway, and so these are mice that lack beta-arrestin. And look what we saw. If you compare that light orange line to the dark orange line, there was no drop in or decrease in uterine contractility in the oxytocin re-challenge compared to the initial challenge. And all of these mice express the same amount of oxytocin receptor within their myocytes. So then if we go back and look at the other component of the oxytocin receptor desensitization pathway, GRK6, and look at the effect in mice that lack that hormone. In these experiments, what we did is we implant this pressure catheter in the uterus of live mice that has the ability to measure, just like in women, pressure generated within the mice. And if you look at the top panel, this is wild-type mice. We implant a pump in their neck that allows delivery of oxytocin, and 3 to 4 hours later, you see an increase in contractile frequency and amplitude. And look what happens in the GRK6 knockout mouse. They generate much greater uterine contractile frequency, force, and they have this elevated basal tone. And what's really interesting is that these mice deliver full-term fetal demise mice. So the mice, they go into labor, the mice are alive, and they die in labor. And I would argue that they're dying in labor because of this enhanced contractile phenotype. And I don't know that much about the placentas of mice, but there may be just decreased oxygen delivery because of that contractile phenotype we mentioned before in humans. So in summary, things that cause oxytocin or are associated with oxytocin desensitization, like prolonged labor or high doses of oxytocin that are utilized, in some women leads to decreases in uterine contractility, which increase the risk for uterine atony. And then on the converse, when these genetically engineered mice that lacked the desensitization pathway, we see this enhanced uterine contractility. So it begs the question, are there individual variations in oxytocin receptor desensitization that could account for these different contractile phenotypes? Because clearly, not all women exposed to high doses of oxytocin have any issues. Some, it's just a small subset. So we went to the literature, and there's this amazing paper that we found. And so the beta-2 adrenergic receptor in the heart is also a G protein coupled receptor that's desensitized by GRK5, just like it's very similar to GRK6. And what they found is that women, folks that have a certain polymorphism in GRK5 do better in the setting of heart failure. And through molecular studies, they found that this polymorphism leads to enhanced desensitization of the beta-2 adrenergic receptor, essentially acting like a genetic beta blockade, and it simulates pharmacologic beta blocker therapy. This is why that they're doing better in the setting of heart failure. So given this precedent where there's genetic variation in this pathway, could there also be a similar precedent in the oxytocin receptor GRK system, which would affect things that we associated with oxytocin dosing and contractility? And such may be the total oxytocin given or the maximal rate needed, the duration of labor, mode of delivery, or uterine atony. Not all women require the same when we're inducing a woman or augmenting a woman. Some women have a normal contractile pattern at 2 milliunits per minute, and some require 30, and you cannot predict it. It's not due to parity, it's not due to BMI, we have no idea. So there clearly is variation in what requires dosing. And so we designed this experiment to determine if genetic variation in the oxytocin receptor GRK6 gene could explain the variation in oxytocin dosing and labor outcomes among women being induced near term. So what we did is we took women who had participated in a clinical study at Duke who had DNA collected, and what we did is we had 482 women who underwent induction of labor at 36 weeks or greater of a singleton gestation, and we genotyped them for haplotype tagging SNPs within these two genes, the oxytocin receptor gene and GRK6. And our primary study outcome was determine the genetic association, or determine the association of genotype with the maximal rate of oxytocin infusion, but then also all of these other secondary outcomes, including uterine atony. So here's a map of the oxytocin receptor. It's located on the short arm of chromosome 3. It has 4 exons, with the last 2 being coding, and we genotyped the women for 18 haplotype tagging SNPs that we identified. And similarly for the GRK6 gene, we identified 7 haplotype tagging SNPs across the length of the gene. So of our 482 women, approximately 70% of them were non-Hispanic Black. The mean BMI was, I can't read there, but 48% were nulliparous, and the mean gestational age of delivery was 38.9. I'm sorry for the cutoff, but so I'm going to present my data here, the p-value, which is right there and just off a little bit. This is for an additive genetic model that controls for, what we did is went through backward selection to determine all these different covariates that could affect the primary outcome, which is listed at the top. And for maximal rate of oxytocin diffusion and the association with these genotypes, we then controlled for these variables that are listed on the bottom of the slide. And if you can see is if you look at that top, at this polymorphism, women with the GG genotype, their mean maximal rate of oxytocin infusion was 10.9. That increased to 13.8 with GT and 14.0 with the TT genotype. So there's clear associations of genotype on how much oxytocin that they received. And then we found there was 4 other SNPs that had similar associations with maximal rate of oxytocin infusion. Did the same thing for the total amount of oxytocin that received. And again, these are all women being induced. So we got rid of the issues with induction versus augmentation. So here we see for total oxytocin dose, at this polymorphism, women with the GG genotype had a mean total dose of 6.8 units, and that increased to 10.4 units with the TT genotype. Duration of labor, this is pretty cool. If you look at women at this polymorphism, the AA genotype, the mean duration of induced labor was 17.7 hours, and that increased to 23.5 hours with women with the CC genotype at this polymorphism. And then cesarean delivery rate, as example, for at this polymorphism, women with the CC genotype, their cesarean delivery rate was 21%, and that increased to 33.3% with the TT genotype. So these are just the kind of gene maps of the significant polymorphisms that we found and where they're located. So the summary from this was that among women undergoing induction of labor, maternal genotype at these two genes influence these oxytocin dosing variables, the duration of induced labor, and cesarean delivery rate. Sadly, though, that because of this small cohort, we had too few cases of uterine atony to determine the association of genotype with this outcome. But again, in this cohort, like we've seen in others, almost all of our cases of uterine atony were associated with long labors and higher doses of oxytocin. And these outcomes do suggest that there probably is some genetic predisposition that affects this contractile phenotype during labor and in the postpartum setting, whether it's your response to oxytocin, your duration of labor, or potentially what you do immediately after delivery. So again, genetic variations in this system affect oxytocin dosing requirements and labor outcomes. Identifying the functional significance of these variations may then allow for not only a personalization of labor management. Right now, what we do on labor and delivery units, at least at Duke, and I'm sure across the country, it's a one-size-fits-all. And clearly, there are differences in response to this drug that we use all day long up on these units. And not only personalization of labor management, but personalization of management of the third stage of labor to help deal with uterine atony. And again, these genetic variations may explain why we see racial and ethnic variations in uterine atony and postpartum hemorrhage, or why some women, we know that a prior history of uterine atony and postpartum hemorrhage increases your risk. Well, your genes may determine that. So in summary, transition of uterine contractile phenotype from a phasic one to a tonic pattern is very important to control postpartum blood loss. Plug the hole, allow your fancy proteins to work. Oxytocin is the mainstay for prevention of uterine atony, and boluses of oxytocin are utilized immediately after delivery to help provide or help facilitate this tonic contractile response. And I would argue that oxytocin desensitization may contribute some risk for uterine atony, and there may be this genetic predisposition based on genetic variations in the genes and proteins that regulate this pathway. So with that, happy to take any questions.
Thank you.
Yes, sir.
So since you're not going to change the genes, what about pharmacologic intervention with beta-arrestin antagonists?
That's a really, really great question. And so what's really cool about G-protein coupled receptors, well, one, they're the largest pharmaceutical target, I think, class. And what's really interesting about them is that they have the ability to have what's called biased agonists. And so they, on a G-protein coupled receptor, beta-arrestin not only promotes desensitization, but also signals independently through cell growth and proliferation pathway. So at any given receptor, you can have an agonist that preferentially stimulates the G-protein pathway, preferentially stimulates the beta-arrestin pathway, or preferentially blocks either one. And so my lifetime goal would be to develop, is there any requests being recorded? In the bear peel. It would be a non-desensitizing oxytocin receptor agonist. The problem with all of that, which then I learned through the GRK6 mice, is that you probably need some amount of desensitization in labor to provide this normal contractile phenotype that allows for gas exchange. And a non-desensitizing agonist is probably not good for normal labor, but I think it would be great for postpartum. And tomorrow we'll talk about carbetocin and what it does and why I think it probably does something different than what Ferring thinks it does. But.
So just a question about the transition to a tonic contraction algorithm. Does anyone have any idea why that occurs? The oxytocin's the same, although you point out that the bolus is really big.
It is,
but in nature, why does that happen?
I have no idea, and the issue is that it happens in women all the time who don't receive oxytocin. It's just a normal thing that happens. The fetus comes out, the placenta comes out, and that thing clamps down and becomes ischemic. It turns that white color that we were. And I don't know. We need smarter people to look at this than nobody knows. I don't think people know, or at least I'm not aware of that.
Is it reasonable to think that it's primarily related to oxytocin as opposed to other factors in nature?
I think that even though I love oxytocin, it's my favorite. After skiing, it's my favorite thing. It does. It's like my brain is on. No, but I think it would be naive to think that it's only oxytocin, it's only desensitization. That it's got to be change in response, and it may be that it's some immediate change that changes as receptivity to all kinds of myosin. I don't know.
So we have a question back here, and if we could ask people to give their names.
I'm Kelly McLean from Vermont.
Yes, ma'am.
So one question, what do we know about oxytocin receptor desensitization in spontaneous labor as a result of endogenous exposure to oxytocin?
Yep,
important that we-
I think it's one of these things that it naturally occurs, whether you're having oxytocin releasing from your brain or whether you're giving synthetic version, which is identical. How do you measure that? Clearly, though, and we'll talk tomorrow, we'll look at the incidence of uterine atony that's increasing over the last 15 years. Clearly, the greatest increase has been in women who are being induced, whether they're delivered vaginally or cesarean. So there's something about induction. Now, is it the thing that caused them to be induced, or is it the drug itself?
Right, and that's...
But I think that there's probably greater degree of desensitization in women who require augmentation than those that don't. But there are clear benefits to induction augmentation. I'll make that very,
It will
very
be
crystal
difficult
clear.
and important to get to that, though, because, right, the conversation on whether induction increases cesarean rates, right, that's been raging,
Yeah, and
controversy
that
for a long
hopefully
time.
we'll
And
find
hopefully
out
we'll find out, but it's the
this
same
year,
issue,
but-
is that what happens, before the big babies, the chorioamnionitis,
Yeah.
that results in the use of the Pitocin. Anyway.
Chad, thanks. That's fantastic.
Okay.
A couple of things that came to mind is that we often decide what we want to do is give a Pitocin administration break in labor to try to see if we can regenerate the contractile pattern. Any thoughts about how that might actually be true and what you looked at? And also, if we're worried about prolonged administration of more than 20 milliunits for an hour for more than 4 hours, would there be a place for taking them off Pitocin to reduce the postpartum uterine atony rate?
It is. There's a lot of funny things that happen on labor and delivery units. And I think that's something that got me interested. There's a lot of just witchcraft that goes up there. And our nurses do all kinds of stuff that they half it and shut it down for an hour. And I don't know. I honestly don't know.
And the other question I had is that we often find that with intrauterine fetal demise, with consumption coagulopathy and disseminated vascular coagulopathy, it's very difficult to get the patients into labor with the use of oxytocin.
Right.
And is there any role for why that might be?
I don't know. We've had a few big cases at Duke of just huge concealed abruptions, and I've been amazed that you cannot get those uteri to contract. But what's really funny, where lower levels of abruption, thrombin is this very potent uterotonic agent, and it causes these very tonic, and maybe it simply is just the presence of blood with shearing of the placenta that leads to that transition to the tonic. But at some point, there is a point where you don't get it to contract, and I don't know why.
Actually, it was about, if you wanted to speculate about the role of thrombin, and I thought that would make a good segue to Dr. Burbank's talk, but I think you've
Yeah, we do know that thrombin is a potent uterotonic agent. When you take myometrial strips and put them in an organ bath and add thrombin, they contract. And I wonder if that's some of the physiology of why we see these women who are abrupting coming in with these tonic contractions. When you give thrombin, or when you block that thrombin receptor, you can reverse some of that contractility. But after that, it's beyond over my head.
Great. Thank you. That was
you very
great.
much.
Very stimulating. Yeah. And now it's my pleasure